Welcome to video 3 of Genetics, the Code Broken. In this video, we're going to be looking at polygenic inheritance and highly variable genes. When we look at polygenic inheritance, we're going to try and understand what it means and describe an example, and we're going to describe this example further in class. And then we're going to look at highly variable genes in DNA and how we might use these for forensics, for paternity testing, and for determining the pedigree of animals. When we look at polygenic inheritance, we're talking about when two or more genes affect a single characteristic, resulting in continuous variation. This is in contrast to multiple alleles, which we just looked at, and that's where there are two or more, when there are more than two alleles, that affect a characteristic. Here we're looking at two or more genes. If there are a greater number of genes affecting a characteristic, there will be a smaller effect of any one gene. And when we look at these, we can look at human height, milk yield in cattle, and egg laying in poultry. When we look at human height in particular, we don't just have tall and short people, we have variations in between. And that's what is meant by polygenic inheritance. When we look at highly variable genes, we're looking at repeat sequences of junk DNA, which is found at specific locations, so defined loci, in the human genome. Individuals vary in these sequences for each allele. And when we can isolate 10 or more variable genes from an offspring and parent, we can compare these. And so that's how we do paternity testing using electrophoresis. Since most of our DNA is identical, it's necessary to use these highly variable genes, DNA, fingerprinting, and so on. And that's where we look at restriction fragment lengths. And we're going to look at that a bit further. So when we do DNA fingerprinting, we're looking at the use of non-coding regions of DNA because they uniquely and in a genetic way identify an individual. By using a DNA marker, it is able to identify the presence of a useful or desirable gene. And then when forensic scientists use 10 of these markers, they can create a DNA profile or fingerprint of an individual. And the chances of having another individual having the exact same sequences is extremely small. So I spoke about, when I was speaking about highly variable genes, the use of electrophoresis. And this is where we use something called restriction enzymes. And these cut DNA at specific points. When each person in the test is their DNA is extracted and the same restriction enzyme is used on all of their DNA, it will cut each fragment depending on the individual into smaller fragments, and then they can be sorted by size by a sieving process of electrophoresis in a gel. When we have more than one sample in here, we can use the patterns that are produced in this electrophoresis, so we can see it through probes or the banding patterns, and we can compare these, and the similarity in the pattern they produce will indicate a similarity in their DNA because this restriction enzyme has found the same specific points in their DNA that has been extracted. We're going to go through this further in class, so make sure you're there. Thank you for watching video 3. Make sure you tune in for video 4.